to today's video lecture. In today's video lecture, I'll be solving previous year's NEET questions and discussing the answers. And this is for the chapter Biomolecules, and we are in part 4 of the series. So if you haven't checked out part 1, 2, 3, please do so. The links will be there in the description box below. Alright, so let's begin. Okay, so question number 1. Which one of the following statements is incorrect? And this appeared in 2015. So it was a cancelled exam, I guess. We have options A, B, C, and D. Let's go through them. Option A, the competitive inhibitor does not affect the rate of breakdown of the enzyme substrate complex. So you define the statement that is incorrect. Okay. Well, A is correct. The presence of the competitive inhibitor decreases the Km of the enzyme for the substrate. Actually, that's incorrect. So B ideally should be the right answer. Let's go through C and D. C, a competitive inhibitor reacts reversibly with the enzyme to form an enzyme inhibitor complex. Yes. D, in competitive inhibition, the inhibitor molecule is not chemically changed by the enzyme. Alright, so if you go through all the options A, B, C, and D, option B is the correct answer because it is the incorrect statement. Right, so your answer is B. Let's go to the discussion. Okay, so if you have gone through NCRT, you should be familiar with this graph on the right over here. And what we see over here is the blue line is the Michaelis-Menten equation graph, which represents enzyme activity versus the concentration of the substrate. Okay, and the red line over here represents the inhibitor. Okay, and how that performs in a situation where you have to compare the enzyme activity versus the concentration of the substrate. Now, if we go back to our question, the right answer is that in the presence of competitive inhibitor, and this is a very, very important information, okay, for you to understand how Michaelis-Menten equation and this graph works. Okay, the presence of competitive inhibitor increases the Km value, okay, whereas the Vmax value remains the same. Always remember, in the presence of competitive inhibitor, the Km value increases, whereas the Vmax value remains the same. Okay, that we can see here. So you see the red line over here, that represents competitive inhibitor. Now this is all done experimentally. Okay, this the blue line that you see here, and you know how this graph was plotted and all, it was all done experimentally in a lab, where they had tested the enzyme and seen how that affects the activity of the enzyme versus the concentration of the substrate. Now it is very important for you to understand what Km means. Okay, what Km means. Okay, and I'm not going to the derivation of the Km value in this particular presentation, a video. I mean, I'll probably do one later on where I'll discuss in much detail how Km value was derived. But to understand what Km is basically the concentration of the substrate when velocity or the rate of the enzymatic activity is half of its maximum. Okay, that means 50% of the enzymes active sites are being occupied. Okay, to reach half of that level, okay, you know Vmax, let's say Vmax was 50. Okay, it's going at a 50 meters per second speed. For example, just taking an example of a car. Now to reach 25, what was the substrate concentration? Okay, that is half. So at that point, at this half Vmax, what was the substrate concentration? Okay, for example, if you have 6 molecules of enzyme here, 1, 2, 3, 4, 5, 6. Okay, you have the active sites present over here, and you have substrate. This is I'm talking about in a normal situation, nothing to do with inhibitors involved. Okay, and we are not even talking about inhibitors as yet. We are just talking about this blue line here. You see this you see this blue line here. We're talking about this graph. We're not talking about the red graph. We're not even talking about the inhibitors as yet. So let's say you have these enzymes present in a solution. You have your substrate molecules here. Okay, now if 3 of the active sites were occupied by the substrate, that means it has, let's say this, I'm just giving an example. That means half of it has reached. Okay, half of the activity has been reached. We're not talking about full capacity of the enzyme activity as yet, we're just talking about half. Okay, so this is how it normally works. So now when half of it is reached, what is the concentration of the substrate? That is the Km value. Okay, so basically this is what it is. Now we have a third character in the story, which is the villain. Okay, and we are talking about inhibitor. So this orange dots over here, they are the inhibitors. Okay, so you have your heroine, which is the enzyme, and there are two guys pining for her. Okay, you have your substrate, okay, and you have your inhibitor here, which is the villain of the story. Okay, so you have three characters now. All right, and now the inhibitor comes into the picture, and the inhibitor also wants to occupy that site, the active site of the enzyme. Okay, it behaves like the villain for the substrate because now it's competing with the substrate. Okay, so even I'm going to occupy that active site. All right, so now the inhibitor begins to occupy these active site spaces over here. Yeah, so when that happens, now this is also again done experimentally. So when they added inhibitor, what they noticed is that the Km value increased. Okay, the Km value in the presence of a competitive inhibitor increased. Okay, whereas the Vmax value remained the same. If you look at the maximum value of the velocity is the same. There's no change in both a normal situation as well as the presence of an inhibitor. Okay, so that means this shows that when substrate concentration is high, okay, higher levels of substrate concentration, the velocity doesn't change. The inhibitor does not have any effect on the velocity, maximum velocity. Okay, so that means the Vmax value remains the same, right? This is a situation where the substrate concentration is high. But when you look at lower levels of substrate concentration over here, the Km value was shown to increase. Okay, the value of Km increased. That means now to reach half of that velocity, the Km value has to increase. That means substrate concentration has to increase in the presence of an inhibitor. That's what it basically means. Okay, so in the presence of competitive inhibitor, if you notice at higher levels of substrate, that means Vmax value does not matter, it does not change. But when you look at lower concentrations of substrate, the Km value increased. All right, if this is the normal, if you see this blue line is the normal, then this red line is after the addition of inhibitor, the Km value has increased. Okay, so now when obviously when there's an inhibitor and there's basically if you look at it logically, the amount of substrate has kind of decreased and the inhibitors are beginning to occupy that space of the active sites, right? So now that you have inhibitors involved in this, so they'll begin to occupy the space there, right? So now you need more substrate present in the solution to at least bring the value to half of the maximum velocity. That is why you see a increase in the Km value. Okay, so that's the logic behind this. You see Vmax remains unchanged, but Km value has to be increased because now inhibitor is beginning to occupy the enzyme active sites and substrate needs to be increased to be able to perform the normal reaction. Okay, so Km value increases in competitive inhibition and Vmax value remains the same. All right, clear? So I guess I've answered the question. If we come back over here to the question, the presence of competitive inhibitor decreases the Km of the enzyme for the substrate, but that is incorrect. It increases the Km of the enzyme for the substrate. All right. Okay, let's move on then. Okay, so question number 2. Select the option which is not correct with respect to the enzyme actions. Appeared in 2014. A, substrate binds with enzyme at its active site. We find the incorrect statement. B, addition of lot of succinate does not reverse the inhibition of succinate dehydrogenase by malonate. Actually it does. So I think B should be the right answer. C, a non-competitive inhibitor binds the enzyme at a site distinct from that which binds the substrate. And D, malonate is a competitive inhibitor of succinic dehydrogenase. Okay, so A, C, and D are correct, but option B is incorrect. Let's go to the discussion. So addition of succinate, which is the substrate, reverses the inhibition of succinate dehydrogenase by the inhibitor molecule. That's malonate. Malonate is one of the inhibitors. So I've already discussed this. So if you have the active site of an enzyme here, and obviously you have two competitors, you have succinic acid, sorry, which is the normal substrate, and you have malonate, which is the inhibitor. So both of them are competing for the same site. Okay, so but if you add more succinate, then it overpowers the inhibitor malonate. Okay, so that then since it overpowers that the inhibitor, it begins to then occupy the enzyme's active sites. All right, so basically this is the logic, and this is competitive inhibition. Okay, we already discussed competitive inhibition in the previous question, so it's the same logic here. It's just that you need to remember this example. Again, this example is very important. Okay, let's move on then. Question number 3. Transition state structure of the substrate formed during an enzymatic reaction is, and this appeared in NEET 2013, A, transient and unstable, B, permanent and stable, C, transient but stable, D, permanent but unstable. So transition state, from the word itself you can identify that it's a transient structure, but it's an unstable structure. All right, so the answer is A. Okay, so in enzymatic reaction, the transition structure is transient and unstable. Okay, so if you have a substrate and you have an enzyme, so there's an enzyme substrate complex formed, okay, before the product. So this structure is transient, it's temporary, it's not permanent, and it's an unstable structure. Okay, it's an unstable structure because it will then either be converted, it will break down again to form the product and the enzyme, or if it's a reversible reaction, will again then break down into substrate and enzyme. Okay, so it is a transient and an unstable structure. Pretty simple to understand, but just remember the nature of this transition structure. Okay, I guess we've answered the question. Let's move on then. Question number 4. The essential chemical components of many coenzymes are, and this appeared in NEET 2013, A, carbohydrates, B, vitamins, C, proteins, and D, nucleic acids. Okay, so the essential chemical components of many coenzymes are actually vitamins. Okay, so the answer is B. I hope you know what coenzymes are. It's the organic non-protein part of the enzyme. The vitamins are derived from vitamins. Okay, so some examples are coenzymes of vitamin B complexes are NAD+, FAD. Okay, so they are all coenzymes of vitamin B complexes. Okay, so yeah, vitamins play a very important role. That's why it is important to eat vitamin rich sources of food, because they form the coenzyme part. Okay, all right, let's move on then. Question number 5. Which of the following statements about enzymes is wrong? This appeared in Karnataka NEET 2013. A, enzymes are denatured at high temperatures. Yes. Enzymes are mostly protein, but some are lipids also. No. B could be the incorrect statement here. C, enzymes are highly specific. D, enzymes require optimum pH and temperature for maximum activity. Yes. So the answer is B. Yes, we know enzymes are mostly proteins. All enzymes are proteins, but not all proteins are enzymes. You know that line. But there's a exception. Okay, it's RNA based protein also called RNA based enzyme, I'm sorry, called ribozyme. Okay, it's an enzyme which is RNA based. So the RNA part of that structure forms the enzymatic part. Okay, which helps in converting a substrate to a product. Okay, so yeah, the enzymes are proteinaceous in nature, or exception is the RNA based enzyme, which is ribozyme. And again, I've discussed this in the previous lecture also, what ribozymes are. So you can go through that. All right, let's move on. Question 6. The curve given below shows enzymatic activity in relation to three conditions: pH, temperature, and substrate concentration. What do the two axes, x and y, represent? Okay, so you have x-axis and you have y-axis. This appeared in 2011. So you have enzymatic activity versus the pH. Then you have B, temperature, and enzymatic activity. You have substrate concentration and enzymatic activity, or enzymatic activity and temperature. So x-axis represents the temperature and y-axis the enzymatic activity. So the answer is B. Actually, it represents pH and temperature. See, these two graphs are taken from NCRT, so you should be familiar with that. So x-axis represents either the temperature or pH, and the y-axis represents enzymatic activity. Okay, so you see that at this peak point, which is the maximum velocity or the rate of the enzymatic activity, and as you go further down the pH, the activity decreases. Similarly with temperature, both of them have similar graphs. That's how they function. That's why we say that at a particular pH and a particular temperature, the enzyme performs very well. Okay. Okay, so please read the graphs that are there in NCRT. Okay, I mean study them. Right, I guess we can move on. Question number 7. 3 of the following statements about enzymes are correct and one is wrong. Which one is wrong? Okay, means 2010. A. Enzymes require optimum pH for maximal activity. We just discussed that. Enzymes are denatured at a high temperature, but in certain exceptional organisms, they are effective even at high temperatures, 80 to 90 degrees, that is. Yes, that's correct. Enzymes are highly specific. Yes. Most enzymes are protein, but some are lipids. We just discussed this. So it's option D, which is the correct answer. Was the incorrect statement here. A, B, and C are correct. Okay, so it's answer B. So the RNA based enzymes such as ribozymes. Now if you go back to the question again, it says enzymes are denatured at high temperature. Yes, we know that, we just saw the graph, right? But in certain exceptional organisms, they are effective even at high temperatures. Okay, so there are certain enzymes that are produced by bacteria or some organisms where they are thermophilic. That means they are heat loving. The thermophilic enzymes. Okay, this is another exception. Just like ribozymes, we see the RNA based enzymes. The thermophilic enzymes also, which perform well at very high temperatures like 80 to 90 degrees Celsius. Okay, so apart from that, yes, the normal graph is that they are denatured at high temperature, and we've seen that in the graph also. But these are exceptional organisms. All right, so they are called thermophilic enzymes. That means they're heat loving enzymes. Okay, let's move on. Question number 8. The figure given below shows the conversion of a substrate into product by an enzyme. In which one of the 4 options A to D, the components of reaction labeled as A, B, C, and D are identified correctly? This appeared in mains 2010. So we have to identify A, B, C, and D, right? Okay, so A is the transitional state. Yeah. No, sorry. Yeah, A is a transition state. Yes. B is a potential energy. C is activation energy without enzyme. And D is activation energy with enzyme. I've already discussed activation energy in the previous lecture. Okay, in much detail. I've discussed this graph also that's given here. So the answer is B. Yeah. So this is taken from NCRT, this graph here. So this should be familiar. And yeah, so basically this represents the substrate and this formation of product. and this is the potential energy on the y-axis, this is the progress of reaction on the x-axis. So now when substrate needs to be converted with product to a product, there is a certain amount of energy that it needs to be able to pass. Like there's an energy barrier, okay? There's an energy barrier and it needs to pass this energy barrier to be converted to a product. So without the enzyme, there's a lot of energy required for this barrier to be crossed over. It's like climbing up a mountain. But if you create a tunnel inside the mountain, that is, I'm giving the analogy of a mountain and a tunnel with respect to without enzyme and with enzyme. So if, just imagine there's a tunnel created here. It's easy to pass from A to point B. Similarly, if there is an enzyme present in the reaction, then the conversion of substrate to product is much easier. There's less energy required to do that. Okay, so that is why activation energy with enzyme, you see this graph here, there's less energy, and without the enzyme there is more energy. Basically that's what it is. All right, but yeah, since this has appeared and this graph is present in NCERT, please study the graph well. Okay, let's move on then. Question number 9. A competitive inhibitor of succinic dehydrogenase is, we already discussed this before, it's appeared in 2008. You have A, alpha ketoglutarate, B, malate, C, malonate, and D, oxaloacetate. Okay, so there are two answers to this question, C and D. Okay, answer is C and D. So malonate is a competitive inhibitor, also oxalic acid, that's oxaloacetate also, and glutaric acid, okay, are other competitive inhibitors. Okay, so please learn the names. We move on. Question number 10. Modern detergents contain enzyme preparations of, and this appeared in 2008, A, thermoacidophiles, B, thermophiles, C, acidophiles, and D, alkaliphiles. Okay, so modern detergents contain enzyme preparations of D, alkaliphiles. Okay, we'll go to the discussion now. Answer is D. Now, detergents that you use at home, they contain a lot of enzymes because they take out that stain, right? So they need, like, you have food stains on your clothes or anything. So you need enzymes to break those stains out. All right. So modern detergents contain enzyme preparations of alkaliphiles. Now, there's a list of the enzymes that are present in detergents. Okay, they're mainly protease, amylase, lipase, cellulase, manganese, and pectin lyase. Okay, these are the list of enzymes that are there in detergents. Now, pH has to be maintained, right? There has to be a particular pH that is maintained with the help of buffers, keeping it alkaline in nature. So these enzymes perform well in that condition. Okay, so the particular temperature that is set, then you have a particular pH that is maintained, and these enzymes need to act on the stain. Okay, so that is it. Yeah, you can note down the list of the detergents that are there. Just understand that, learn it as a fact. Then modern detergents contain enzyme preparations of alkaliphiles. Okay, they are alkaline loving. Okay, let's move on then. Question number 11. An organic substance bound to an enzyme and essential for its activity is called, and this appeared in 2006, A, isoenzyme, B, coenzyme, C, holoenzyme, and D, apoenzyme. So organic substance bound to an enzyme is coenzyme. Okay, so the answer is B. And coenzymes are non-protein organic cofactors that are loosely bound to the enzyme. We already discussed coenzymes, different types of cofactors that are present. Okay, that are attached to the apoenzyme. Let's move on then. Question number 12. The catalytic efficiency of two different enzymes can be compared by, this appeared in 2005, A, formation of the product, B, pH of optimum value, C, Km value, D, molecular size of the enzyme. Okay, so if you've given two different enzymes, then you have to compare the catalytic efficiency of those two particular enzymes. We already discussed this in the first question also. How would you compare? On what basis? So, Km value is the right way to compare two enzymes based on the catalytic efficiency. Okay, answer is C. Let's go to the discussion. Okay, so I've already discussed Km. Km, or Michaelis Menten constant, is defined as the substrate concentration to produce half of the maximum velocity. We can already see that from the graph here, right? That means half of the enzyme molecules are occupied by the substrate, and Km is a way of measuring the strength of the transient complex. Okay, so we know that the transient complex is formed, right? Now, I already discussed how these enzymes are present, right? And substrate occupies the active site of the enzyme, correct? So, if we want to see how much of the substrate is occupied in the enzyme active site when this maximum velocity reaches half, okay, then basically you have to do an experiment. You have to do a series of experiments where you get different plot points, and you plot the graph over here, velocity versus the substrate. Okay, you plot this graph, and then you can find half, Vmax already, yeah, from this. Now what will be Vmax by 2? This is 50, this will be around 25, right? And then you can find the substrate concentration over here. Now this substrate concentration will be Km, right? This substrate concentration will be Km, and this is done only for one type of enzyme. Okay, now you have another enzyme, and you do the same experiment again, and you plot the different plot points, enzymatic activity versus a substrate, and again you get the answer, you get some Km value, okay, from that particular graph also for that particular enzyme. Now when you compare the two, how would you know which one is more efficient, which is less efficient? How would you guess that, right? So low Km value represents strong affinity between substrate and enzyme. Okay, so low Km value represents strong affinity. This is a very, very good important point to remember when you're studying enzymes. Okay, now let's say you have another graph, some enzyme you have done, and you have to check the efficiency, and you are comparing. Okay, you found a Km value which is lower than this. Okay, you found a Km value which is lower than the first enzyme one. Now how would you say that this, which is efficient, which is not efficient? We can say that the lower the value of Km, the more stronger their affinity is, the substrate has towards the enzyme. That means there is less amount of substrate that is required to reach that half of the maximum velocity. Okay, less substrate is required for that enzyme to reach half of its maximum velocity. It doesn't have to do much work. There is more attraction, there's strong affinity. That means there is a stronger attraction between the substrate and the enzyme when there's a low Km value. Just imagine if the Km value was here. Okay, you had another third enzyme. Okay, you have a third enzyme, you did the experiment, you got different plot points in the lab, and you plotted the graph. Okay, you plotted a graph over here, and your Km value reached over here. I'm sorry, the Vmax value reached Vmax by 2, that is reached over here. That means it needed more substrate to reach even half of that maximum velocity. Okay, that means there is less affinity for the substrate towards the enzymes. Okay, so a high Km value represents weak affinity between substrate and enzyme. That means that substrate and their enzyme have to do more work. Okay, that means they are not very attractive to each other. Whereas a low Km value represents strong attraction between the substrate and the enzyme. Okay, there's less amount of substrate that's required to even reach half of that maximum velocity to be able to convert into a product. Okay, so that means that it represents strong affinity, whereas a high Km value will represent a weak affinity between the substrate and the enzyme. That means more amount of substrate is required to even reach half of that maximum velocity of the enzyme. Alright, so this is the difference between the two, and that's how you can compare different enzymes. Okay, when you're doing a series of reactions or you've been given enzymes, and you have to check the efficiency of those enzymes. Let's say you have 5 enzymes, you can even do this experiment for 5 different enzymes and then compare all of them and see how it is. In this case, obviously, according to the question, there were 2 enzymes given. So you go to a lab, you test that enzyme, you find the Vmax value or the V value for different substrate concentration, and then plot the graph and find Vmax by 2 to find the Km value. You find the Km value for one particular enzyme, you do the same for the second enzyme, and then you compare the two. So all this has been done experimentally. That's how you have this Michaelis-Menten equation, which helps you understand how enzymes function. Okay, so this particular information fact is very, very important to understand enzymes, okay, and how they function. So yeah, the catalytic efficiency of 2 different enzymes can be compared by the Km value. All right. Yes, I've answered the question. Okay, so question number 13. Which one of the following statements regarding enzyme inhibition is correct? Here's another question based on inhibition. This came in the year 2005. A, competitive inhibition is seen when a substrate competes with an enzyme for binding to an inhibitor protein. So to find the correct statement. Okay, with an enzyme for binding to, it doesn't compete with an enzyme. So A is incorrect. Competitive inhibition is seen when the substrate and the inhibitor compete for the active site on the enzyme. It's true. C, non-competitive inhibition of an enzyme can be overcome by adding large amounts of substrate. No, that's not the case. D, non-competitive inhibitors often bind to the enzyme irreversibly. No, there are reversible reactions also. So, option B is the right answer here. Answer is B. Now, I've already discussed competitive inhibition. I'm not going to discuss this again. So this diagram basically shows what competitive inhibition is. So you have a substrate and a competitive inhibitor, and both of them are competing for this active site here. Okay. So it's when the substrate and the inhibitor compete for the active site on the enzyme. Okay. Clear? Let's move on. Okay, so question number 14. Enzymes, vitamins and hormones can be classified into a single category of biological chemicals because all of these appeared in 2005. A, help in regulating metabolism. B, are exclusively synthesized in the body of a living organism as at present. C, are conjugated proteins. And D, enhance oxidative metabolism. So you have enzymes, you have vitamins, and you have hormones. Then they can be categorized or classified into a single category because all of these, an appropriate answer here would be help in regulating metabolism. Okay, so the answer is A. Now there are different enzymes that participate in the cycles that are present in the cells which help in regulating metabolism. We have vitamins which help in regulating metabolism, and you have hormones also like glucagon, insulin. You have different other hormones like thyroid hormones. Now they all participate in helping in regulating the metabolism. There are vitamins also which help in regulating that, and enzymes also, amylase, lipase, so on. So yeah, the appropriate answer here would be that they play a role in regulating metabolism, and this is a very generic question. We'll just choose the appropriate answer from the options. Okay, let's move on then. Question number 15. In which one of the following enzymes is copper necessarily associated as an activator? This appeared in 2004. A, carbonic anhydrase. B, tryptophanase. C, lactic dehydrogenase. And D, tyrosinase. Okay, so copper is necessarily associated as an activator in the enzyme tyrosinase. Okay, please remember this as a fact. The answer is D. So we know what an activator is the metal ions, which are inorganic, also known as activator, which attach to the active site of enzymes and enhance enzymatic activity. So tyrosinase is one such enzyme, which is copper containing oxygenase, okay, and is used in production of tyrosin, which is the precursor for melanin. Okay, so tyrosin helps in the production of tyrosinase, I'm sorry, helps in the production of tyrosin, and then which is obviously the precursor for melanin, okay, which eventually forms melanin. Okay, and tyrosinase, please remember this as a fact, is a copper containing enzyme. All right. Yeah, let's move on. Yes, this is factual based to remember the fact. Okay, question number 16. Role of an enzyme in reaction is to or as. It's appear in 2000. A. Decrease activation energy. B. Increase activation energy. C. Inorganic catalyst. D. None of the above. So role of an enzyme is to decrease the activation energy. Okay, so the answer is A. We already discussed activation energy before, and yeah, I've already discussed this before, so it's like the same thing. So the role of the catalyst is to lower the energy barrier of the reactants, hence allowing the reaction to go faster. Like you want to go from A to B, so you have the activation energy with enzyme is definitely lower, okay, as compared to the activation energy without enzyme. So if we go back to our question, yeah, so decrease the activation energy. This is basically the whole premise of the use of enzymes. Okay, let's move on then. Yeah, in biological system, the enzyme increases the rate of the reaction temperatures below 40 degrees. I've already discussed this previously when I discussed activation energy, so it's a similar answer to that. But just to understand what enzymes actually do. Okay, question number 17. Which factor is responsible for inhibition of enzymatic process during feedback? This appeared in 2000. Okay, so which factor is responsible for inhibition of enzymatic process during feedback? A. Substrate, B. Enzymes, C. End product, and D. Temperature. So the factor that's responsible during feedback is actually end product. Okay, we'll discuss to what feedback inhibition and feedback regulation is. The answer is C. Okay, let's go to the discussion. Okay, so when you talk about feedback and when the end product plays a role in inhibiting a particular reaction, there are two types: feedback regulation and feedback inhibition. Okay, so let's look at what feedback regulation means. It's the accumulation of end product which will decrease the synthesis of enzyme. This is called negative feedback regulation. So let's say if you have a reaction, you have a substrate A which produces a product B, and then C, and then and then E. So every reaction has an enzyme which participates in this. Okay, so that means substrate A forms product B, and then forms product C, and then D, and then E. Okay, and then every reaction there is an enzyme involved, right? Now when there is too much accumulation of E, it will immediately go and stop this reaction. Okay, now this happens usually in cells. Okay, it's a way of regulating the energy that is required to produce a particular product. So if a series of reactions are taking place and the last product, that's E, and there's accumulation of that product E, it will go and stop this first reaction. Okay, it will stop this first reaction, so all these products will not be formed because there's no need for it, because already there's too much of product E already formed. So this is called negative feedback regulation. Now feedback inhibition or end product inhibition is when the end product allosterically inhibits the enzyme. Okay, so for example, that you see here, okay, you have the end product. Now carbamoyl phosphate and aspartate together eventually form CTP, cytidine triphosphate. Okay, this is a reaction that's happening, and CTP is the final product produced. Now, when there's too much accumulation of CTP, CTP itself will go and allosterically attach to aspartate transcarbamylase, and which will stop the reaction from happening. Okay, this is end product of feedback inhibition. Okay, there's feedback regulation, there's feedback inhibition. So allosterically is when this CTP binds to this ATC, that's the enzyme ATCase, in this particular reaction, to stop the production of further CTP from happening, because we don't need CTP. So there's feedback regulation, there's feedback inhibition. Both the cases, the end product plays a role in stopping the reaction from happening. All right. So these are the two types. Oops, sorry. Let's go back to our question. A question said, which factor is responsible for inhibition of enzymatic process during feedback? Now in this case, it's neither substrate, enzyme or temperature. It is C, end product. Okay, C is end product. Okay, let's move on to the next question. Question number 18. Enzymes are not found in, this appeared in the year 2000, A, fungi, B, algae, C, virus, and D, cyanobacteria. So enzymes are not found in C, virus. Okay, in fungi, algae, and cyanobacteria they are found. They are absent in virus. Okay, so this is a factual based question, so you can answer that quite directly. Question number 19. Cofactor prosthetic group, they're talking about prosthetic group, is a part of hollow enzyme. Now it is A, loosely attached organic part, B, loosely attached inorganic part, C, accessory non-protein substance attached firmly, D, none of these. Okay, and this appeared in the year 1997. So we looked at different cofactors, right, previously, the definitions of the different types of cofactors. And in this case, they're particularly talking about prosthetic group. And it is an accessory non-protein substance attached firmly. Okay, so the answer is C. All right. So prosthetic group is not protein organic factor that is tightly bound to the enzymes. The coenzymes are non-protein organic factors that are loosely bound to the enzymes. Okay, so these are the two different types of cofactors, prosthetic group and coenzymes. All right, so we've already discussed this before. Can move on to the next question. Question number 20. Which is a typical example of feedback inhibition? See, now you see the term inhibition and they put that in quotes. Okay, and this appeared in the year 1996. A, cyanide and cytochrome reaction. B, sulfur drugs and folic acid synthesizer bacteria. C, allosteric inhibition of hexokinase by glucose 6-phosphate. And D, reaction between succinic dehydrogenase and succinic acid. Okay, so clearly it is answer option C. Okay, which is the correct answer. We already discussed this previously, the difference between inhibition, negative feedback inhibition, and feedback regulation. Okay, the end product inhibition and a feedback regulation. That's so allosteric inhibition of hexokinase by glucose 6-phosphate. Okay, it's a typical example of feedback inhibition. So the answer is C. Sorry, let's go back. So yeah, here you see a flow chart of glucose being converted to glucose 6-phosphate and in the presence of hexokinase. Alright, so glucose 6-phosphate allosterically inhibits the function of hexokinase. And this is an example of feedback inhibition. So if you look back at the options, yeah, it's clearly C. Okay. So now you know the difference between regulation and inhibition, feedback inhibition. So this is a very important difference to note, right? Okay, so question number 21. Enzymes have slightly different molecular structure but performing identical activity are, and this appeared in the year 1991, A, holoenzymes, B, isoenzymes, C, apoenzymes, and D, coenzymes. So enzymes having slightly different molecular structure but performing identical activity, and they are clearly B, isoenzymes. So the answer is B. Okay. So now there are multiple forms. In nature, there are multiple forms of the same enzymes, okay, occurring in the same organism and having similar substrate. Okay. There are some examples such as this A is alpha amylase of wheat endosperm, has 16 isoenzymes. I mean, there are multiple forms of the same enzyme. Okay. LDH has 5 isoenzymes. Alcohol dehydrogenase has 4 isoenzymes in maize. So these are different types of examples of isoenzymes found in nature. Clearly, it's a slightly different molecular structure but performing identical activity. Okay, clear? This was simple, direct. Okay, so we come to the end of this video lecture, and I hope you've enjoyed today's video lecture, and I hope you've learned something new today. So don't forget to like, share, and subscribe to MG NEET Biology, and I'll see you in the next video lecture. Right? So bye.